Hello, my friends, and welcome back to another exciting unit here in online biology. So, this week we are going to continue our investigation into learning more about the complex interactions that the natural world provides for us, so that way we can study it as biologists. In the last few weeks, we learned about cell division. We learned about mitosis and meiosis, how mitosis wants to produce genetically identical cells, and meiosis is geared more towards sexual reproduction. In another lesson, we learn more about the transcription and translation process, how DNA starts to replicate itself before either mitosis or meiosis needs to begin, and how the combinations can produce amino acids, which eventually can help us develop proteins, and then some of those proteins can contribute to the formation of new genetic uniqueness that keeps our planet and our biosphere diverse. So this week, I want to take you on a journey. I want you to learn about this topic in the light that it shows us why we have genetic diversity. And to set the stage, I want to tell you a little bit about a historical narrative and how we started to think about this concept, because it's a relatively new concept when we look at science. The year is 1895. The place, Europe. Europe back then was struck by a famine. This famine may have been induced by humans. The story goes that the church in a small rural village of Europe was noticing a problem. The local clergymen were not being able to be working the fields as often. The bishop, or the one that oversees all the clergymen, asked and hypothesized, because they did follow science and heavily acknowledged scientific concepts back then. But he noticed this change. He wrote a hypothesis as part of the scientific method and asked the question: why was the clergymen not performing their tasks the way they should? And so he went out and he conducted a survey and was like, hey, why are my clergymen not being productive at their jobs? Why are they not transcribing notes and translating ancient texts? Or why are they not working the fields to produce the food? And all the clergymen were like, we are hungry. We want to eat good food. And so the bishop went and talked to some of his clergymen that were very fluent in science and pursued it as a hobby. And one such individual was a friar. His name was Gregor Mendel. And soon he would be called the father of modern genetics, because he stumbled upon an idea. He took an observation of the natural world and he looked at pea plants. Peas were actually a very common food source back then because they were easy to grow and preferred cooler temperatures to grow and become productive. And he started to look at the pea plants and realized that some of the pea plants were not producing food. They were not producing enough of it to feed and sustain those that worked for the church. And so he started to document. He started to look at populations of the farm's pea plants and noted a couple of characteristics. He found that pea plants that produced a lot of peas and were very successful were of one color and taller, whereas those that were not being successful were a different color and were shorter. And so he took those two characteristics and labeled one dominant and one recessive. And this is the start of Mendelian traits. And this is what we're going to be learning about this week, is the patterns of inheritance that Mendel started to document and utilize. And I actually replicated Mendel's experiment right here, but instead of using pea plants, I'm using the monocot plant, the common corn. So right here in this tray, we actually have a population of the common corn plant. And we can take a look at some of these corn plants. We've got some tall corn plants right through here that are dark green. And then we also have some shorter white leafed corn plants. In fact, I'm going to prune this leaf off just to show and allow us to compare the traits. So when we take a look at these leaves, this would be the dominant, and then this would be the recessive. And so when Mendel was actually doing his study, he wanted to be able to successfully manipulate and allow for the green plants to become more successful than that of the white plants. Phase the white plants out of the gene pool and allow the green plants to become much more successful. And he was highly successful in doing that and actually ushered in a new concept found in the world of biology. And so that's what we're going to be working on today, is we are going to learn more about this concept. We're going to make identifications of what would you define as a dominant characteristic, what would be a recessive, and then also investigate weird changes that may occur within the gene pool, such as maybe even the production of odd combinations, what they call maybe incomplete dominance, where you'll actually get both traits showing up within a individual. But when we actually take a look at these dominant and recessive traits, we're also this week going to learn on how we can communicate and provide evidence on what you define as the dominant trait and which one is the recessive. And how we can do that is by utilizing a diagram called a Punnett square. So a Punnett square is a way for us to show the percentages of traits within a population, but also what traits you would define as your dominant and recessive. And we can do this by utilizing letters and developing a grid system. I've actually got a Punnett square. I can make one right here. This is going to be a standard 4 square, and we can actually list out our different traits. So I will pick one plant here. This will be the male plant, and then this will be the female plant. And then we're going to actually take the male and the female and convert it into, again, a model we can study by just lining up the dominant and recessive traits. And so we can take the characteristics found in both the male and the female, build a model around it, and then determine what the likelihood of dominant traits passing from one generation into the next, as seen by the blue. And we can mathematically support this by doing a little bit of statistics and answer questions of what is the likelihood of the dominant trait being passed from one generation to the next. Well, we have 3 to 1. So there you go. We have a 3 to 1 chance that our dominant trait, or what we're going to be calling the dominant trait in this problem, has a 3 times likelihood to being passed to the next generation. All right, so if you have any questions about this week's topic, being able to calculate for the dominant and recessive traits is very helpful for going to be our later units in the course, because it's the dominant trait that is reflected through genetic changes that makes an organism more successful. And so later in the class, we're going to learn about topics like evolution and how do those traits actually show up in the organism, actually links us to not just our Mendelian genetics, but also our concepts found in cell division. Another interesting fact is that Mendel was ahead of his time. He not only figured out genetics on the most basic level, but also started to usher in the concept of biotechnology, which is a future topic here in this course, where we can actually take the genetic information of plants and animals and manipulate it using human impacts such as technology to be able to make the changes on the genetic structure to make an organism much more successful, or give it properties that can be utilized in other products such as medicine and agriculture. If you have any questions about this material this week, please reach out. You can contact me using video chat requests, you can send me an email, or even just swing by my office on campus. Thank you and have an excellent day.